Chapter 27 is on bacteria and archaea. So now we're getting into our domains and looking at our taxonomy. And this is where we're starting to get our characteristics that places organisms into the classifications that they fall into. Prokaryotes live everywhere, so we're going to see them in places that are too hot, too cold, too acidic, too salty for most other organisms to live. Now keep in mind that these prokaryotes are microscopic, but there's a lot of them. So there's more than a handful of these guys. Let me back up. In a handful of soil, there's more prokaryotes within that handful of soil than the number of people who have ever lived on Earth. So that is just amazing. We are dividing up our prokaryotes into two domains, bacteria and archaea. Let me get my highlighter going here. We don't have our color straight. There we go. Okay, so two domains, bacteria and archaea. We're going to spend most of our time on the bacteria. So our first concept, structural and functional adaptations are going to contribute to the prokaryotic success. So what you're writing down in your notes here are characteristics that place organisms under a prokaryotic heading. Okay, so we're going to divide these up into bacteria and archaea here in a little bit, but these are general characteristics that define the prokaryotic organisms. So it's thought that the very first organisms were probably prokaryotes. Most prokaryotes are unicellular, but we can see some species that form colonies. Most prokaryotic cells are between 0.5 and 5 micrometers, and it mentions here that's much smaller than the 10 to 100 micrometers of the eukaryotic cell. So what you're writing down in your notes, prokaryotes are small, and then in parentheses put 0.5 to 5 microns. Prokaryotic cells have a variety of shapes. The ones that we looked at in lab are the most common. Those are the spheres, which are the cocci, the rods, which are the bacilli, and the spirals, which come in a couple of different forms, but they are the curlicue-looking organisms. Here's a picture of each of these. So here's the cocci, the bacilli, and the spiral-shaped. We know that all cells have plasma membranes, but prokaryotic cells have cell walls on the outside, and these cell walls contain peptidoglycans. Okay, and we talked about this in lab, peptidoglycans. So this is sugar molecules that are cross-linked by polypeptides. So we've got sugar molecules that are cross-linked with proteins, okay, that make up this. Peptido refers to the protein, the glycans referring to the sugar. Now, we mentioned that eukaryotic cell walls are made up of different materials. The plant cell walls are made up of cellulose, and fungal cell walls are made of chitin, okay? So one of the importance of the cell wall is to help maintain cellular shape, protecting the cell just in general from the outer environment, and it also prevents it from bursting in a hypotonic environment. So that's when water rushes into the cell, and that cell wall is going to prevent it from dying. Archaea, the members of the domain archaea, contain polysaccharides and proteins, but this particular domain does not have the peptidoglycans. So the peptidoglycan is specific for the domain bacteria, not the domain archaea. One of the ways that scientists can start to classify an unknown bacterium is to use the Gram stain. And gram-positive bacteria will stain bacteria purple that have large amounts of peptidoglycan. Gram-negative bacteria will stain pink when they have less peptidoglycan in that outer membrane, which is very high in lipid content. Here's a schematic that's very similar to what we saw in our lab book. So here in the middle, we've got a mixture of gram-positive and gram-negative bacteria. So the purple is the positive, the pink is the negative. And a schematic here showing the peptidoglycan layer of the gram-positive bacteria. Here's the peptidoglycan layer of the gram-negative with that extra layer of the lipids on the outside. A lot of antibiotics will target this peptidoglycan cell wall, and it will actually damage that cell wall. So this is very fortunate for humans because we do not have the peptidoglycan. So antibiotics that target the peptidoglycans will leave eukaryotic cells alone. Gram-negative bacteria tend to be more antibiotic resistant, and that's typically due to an outer covering called a capsule. So typically, if a bacterium has a capsule, it's going to be a whole lot more difficult to kill. This is showing you the bacterial cell wall. This is showing you the inside, and we can see the bacterial capsule on this one. So this particular bacterium is attacking the tonsils because we're looking at a tonsil cell here. So probably leading to the formation of tonsillitis. Some prokaryotes have structures that are called fimbriae. And let's highlight that, fimbriae. And these are extensions that help them to stick to the substrate where the organism lives and feeds, and it can also help them stick to other individuals within the colony. Pili, such as sex pili, may or may not be present. These are longer than the fimbriae, and they will actually allow the prokaryotes to exchange DNA from one cell to the next. This is a figure that's showing us what the fimbriae look like, so these long extensions. So these will help this particular bacterium actually stick to where it lives, or even stick to other bacteria that live in the same area. Some bacteria are modal, so they can actually move in relation to a stimulus. Chemotaxis is moving away from a chemical stimulus. There are other taxes as well where the bacteria could even move away from, say, the presence of light. Modal bacteria are going to propel themselves by flagella. The flagella of bacteria, archaea, and eukaryotes are all different from each other, have a different structure, have different proteins, and it's thought that they have evolved independently of one another. You guys don't have to know the structure. The book goes into the actual structure itself, but we're not going to spend time on that. Okay, so here's what the flagella look like coming off of the prokaryote, and this is the mechanism of what it looks like, and we're not getting into that. Okay, so just to go over some of the structures that we talked about, here again we have flagella. There's the fimbriae that are coming off of the prokaryote. We have a capsule present in this particular case. There is a cell wall that's underneath the capsule, and then there's going to be a plasma membrane on the inside. This is showing you an extra long extension, and this particular extension is the sex pilus. Circular chromosome present on the inside. Remember, we've mentioned before a single circular chromosome present in the nucleoid region. So for internal organization in DNA, we know that prokaryotes do not have membrane-bound organelles. They can, however, have infoldings of the plasma membrane. This is showing an infolding of a respiratory membrane used for making ATP, and here is an infolding of a thylakoid membrane for a photosynthetic prokaryote that is going to undergo photosynthesis. Again, these are not true organelles, but rather they are enfoldings of the plasma membrane itself. The prokaryotic genome has less DNA than the eukaryotic does. The genome is circular, so one circular chromosome. This chromosome is present in the nucleoid region, so there's not a membrane-bound nucleus that are present. There's also some species of bacteria that have small rings of DNA called plasmids that may exist, and these plasmids can be exchanged from one prokaryote to another. Okay, so this is showing you all of these white lines that we're seeing here. This is the chromosome. These little circular pieces of DNA here, these are plasmids. So all of this here is connected into one big circle. It kind of folds in on itself, and then you can see exactly how small the plasmids are. There's another one right there. Now, there are differences between how DNA makes copies of itself between prokaryotes and eukaryotes. So these differences allow us to use those antibiotics and prevent the antibiotics from killing our cells. Okay, so very useful in getting rid of those bacterial infections. But what you need to take home from this slide is that how the cell makes a copy of itself is different in prokaryotes versus eukaryotes. Okay, so reproduction and adaptation. Prokaryotes reproduce by binary fission. They can divide every 1 to 3 hours, and in fact the fastest bacterium is going to divide about every 15 to 20 minutes. So that's pretty, wow, that's pretty fast. So some key features here of our prokaryotic reproduction. These guys are small, we know that already. They're going to reproduce by binary fission, and they have very short generation times. So they go from one generation to the next very quickly, and like we said here, every 1 to 3 hours. Some prokaryotes form what are called endospores, and these endospores can remain viable in harsh conditions for, it says here, centuries. So they can live and persist for very long periods of time. This is what an endospore looks like. So here is the bacterium itself, and this particular bacterium has formed an endospore, and that endospore is going to put a nice coat on the outside, and what's going to end up happening is this bacterium is going to die, but the endospore will persist, and it will stay in this state until environmental conditions are good for that endospore to become a bacterium again. Because the bacterial generation time is very short, this allows prokaryotes to actually evolve fairly quickly. It says here, adaptive evolution in a bacterial colony was documented in a lab over 8 years. So adaptive evolution. So we're looking here at something that Darwin didn't think you could see during your lifetime. And you can see this within 8 years, which isn't even, what, 1/10 of our lifetime? So that's pretty amazing. The next statement's kind of important. It says prokaryotes are not primitive. They're highly evolved. So don't get into your head that eukaryotes are better than prokaryotes because that's not necessarily true. Prokaryotes have to do in one single cell what we do as a eukaryote in many different cells. So they're actually very advanced for what they do. Okay, so that's it for the characteristics, mostly for bacteria itself, but characteristics in general are prokaryotes. Let's talk a little bit about genetic variation in these prokaryotes. So this is concept 27.2. The statement here is that prokaryotes have considerable genetic variation. Okay, so there's 3 factors that are going to contribute to this genetic diversity, to this genetic variation. So number 1, rapid reproduction. and we've already talked about the rapid reproduction. Number 2, mutation. And number 3, genetic recombination. Okay, so you guys know what the rapid reproduction is. You know about mutations already. We've been talking about that as the semester has gone along. Genetic recombinations we've mentioned in lab, but we haven't touched on yet. So rapid reproduction and mutation. So remember, binary fission is how prokaryotes reproduce. This is a type of asexual reproduction, so the offspring cells are identical. Mutation rates are typically very low, but when you couple mutation rates with this rapid reproduction, then those mutations can accumulate fairly rapidly within a population. So over that span of 8 years, you can accumulate a lot of mutations. So the high diversity from mutation actually allows for that rapid evolution to take place. The genetic recombination, now this is when we have DNA and we combine that DNA from 2 sources. And the combining of the DNA is going to contribute to diversity. So for animals like humans, genetic recombination is completed when we undergo sexual reproduction. Okay, we know that there's additional genetic recombination when meiosis takes place. Okay, but the prokaryotes don't get the chance to do this. Right? So they don't undergo sexual reproduction and they don't undergo meiosis, so they have to get genetic recombination a different way. And there's 3 different ways that this can occur. Click on that again. Transformation, transduction, and conjugation. So transformation, transduction, and conjugation. All 3 of these are considered to be horizontal gene transfer. So that's just movements of genes from one species to the next or from one organism to the next. Conjugation, right here, this is when genetic material is transferred between prokaryotic cells, and this is done by sex pilus. Okay, so there's a piece of DNA that's called the F factor that's required for the production of pili. Okay, so you have to have that F factor present. If you have the F factor present, then the formation of a sex pilus can happen, and that sex pilus is going to happen between one bacterial cell and another, and we're going to get the transfer of this F factor. So the F factor is present as a plasmid, and that F factor can be transferred during conjugation. So we've got a figure for this. So here we've got the F plasmid. Here we have the bacterial chromosome. Down here we have the recipient cell. Okay, so this F plasmid can make a copy of itself and take that copy and run it through the sex pilus into the recipient cell and reform itself. Okay, so now we have the F plasmid that has gone from one cell that had it to another cell that didn't. So we've got a transfer of genes on whatever is on that plasmid to a cell that didn't have it before. This is conjugation. There are other types of plasmids. R plasmids is another example of another type of plasmid, and R plasmids can carry antibiotic resistance. So the exchange of R plasmids is not a good thing for humans because it makes these bacteria resistant to a lot of our common antibiotics. Okay, let's back up just a little bit because we need to talk about, we've got conjugation, transduction, and transformation. So let's talk about those 2. Transformation is the ability of the bacterial cell to take up DNA from the environment. So transformation is the ability of the bacterial cell to take up DNA from its environment. We actually use transformation within the laboratory, so it's actually kind of cool to see. It's fairly easy to do a transformation. Transduction is a little bit different because with transduction you are moving DNA from one organism to the next via a virus. Okay, so transduction is introduction of DNA from a virus. Okay, so transformation, you bring in DNA from the environment. Transduction is introduction of DNA by a virus. Okay, let's go back to where we were. Okay, next concept. So diverse nutritional and metabolic adaptations have evolved in our prokaryotes. So this is looking at different modes of nutrition for our prokaryotes. Okay, so this is how these organisms obtain energy as well as carbon, and both of these are necessary for life. So we've got phototrophs and chemotrophs. Phototrophs obtain energy from light, and chemotrophs will obtain energy from chemicals. Okay, photo referring to light, chemo referring to chemicals. Autotrophs, organisms that make their own food, require carbon dioxide as a carbon source. Heterotrophs, which eat their meals, so to speak, these require organic nutrients in order to make the organic compounds. Okay, so they can't get their they can't get their carbon source from CO2. They have to have something that contains carbon in a different way. You have this, so energy and carbon sources are combined to give us our four major modes: photoautotrophy, chemoautotrophy, photoheterotrophy, and chemoheterotrophy. Okay, so those are the four major modes of nutrition, and we're just combining the terms that we saw before, and you have a very nice chart in your book that sums everything up for you guys. Okay, so photoautotroph, light's the energy source. Carbon source for the most part is CO2, and the types of photoautotrophs would be photosynthetic prokaryotes. So on and so forth. You guys can read the rest of these. But this is a nice chart to study. Oxygen is very important to a lot of prokaryotes, but there are prokaryotes who can live without oxygen. So prokaryotic metabolism, how things work within the cell, is going to vary with respect to whether or not oxygen is present. There are obligate aerobes, there are obligate anaerobes, and there are facultative anaerobes. Now, obligate aerobes require oxygen in order to undergo cellular respiration. Cellular respiration is making ATP. Okay? Obligate anaerobes cannot live in the presence of oxygen, so oxygen is toxic to them. So they use an alternative way to make their ATP. Facultative anaerobes can go both ways. So depending upon environmental conditions, they can live in the presence of oxygen, or they can live in the absence of oxygen and take care of things in either environment. There's also nitrogen metabolism. So you need nitrogen because nitrogen is a component of amino acids and also our nucleic acids. So prokaryotes take care of nitrogen in a couple of different ways. One is by nitrogen fixation. So some prokaryotes can actually change the nitrogen that's present in the air to a form of nitrogen that they can use. Okay, so that's nitrogen fixation. You don't have to know going from what to what, but you're going from nitrogen that's present in the air to one that the prokaryote can actually use. There are times when there's cooperation between prokaryotes that allow them to live in environments that they may not be able to live in on their own. So the example that they give here is Anabina, and we looked at Anabina several times in lab. And most of the cells of Anabina are photosynthetic. But there's also nitrogen-fixing cells that are called heterocysts that help exchange these metabolic products such as nitrogen. So it can fix the nitrogen and then pass the nitrogen out to the vegetative cells, and the vegetative cells will then take care of food that the heterocysts will need to exist. Okay, so here's our vegetative cells, right? Those are photosynthetic, and here's the example of our heterocyst that's undergoing the nitrogen fixation. In some prokaryotic species, you can get the same type of metabolic cooperation that occurs in colonies that form a surface, and these are called biofilms. So I don't really want you to know a whole lot about biofilms, but I do want you to know that they exist because biofilms are being used more and more in a lot of different capacities. Okay, on to the next concept. So molecular systematics is illuminating our prokaryotic phylogeny. So you thought you were done with systematics, but we're going to see it popping up over and over and over again throughout the chapter. So this is a big part of Bio 7. So you have to understand the concept. So until the late 20th century, we looked at our prokaryotic taxonomy completely based on what the bacteria looked like, what they did, things that we could see. But now that we apply molecular work, looking at the DNA sequence, looking at the proteins that are made, things have changed quite a bit. So our taxonomy's changed a little bit. So this led the scientific world to get rid of that kingdom Monera and to make the domains. And one of those domains would be domain bacteria and the other domain Archaea. So here we have our universal ancestor, and that universal ancestor gave rise to, in essence, two subsets. Okay, so we've got one branch that's going to lead to the domain Eukarya and the domain Archaea, and the other branch down here is going to lead to the domain Bacteria. Okay, so here we've got the eukaryotes, different members of the domain Archaea, and different members of the domain Bacteria. Okay, they talk a little bit here about PCR. We're not getting into that. We've already talked about a handful of soil could have so many different species. And in the last chapter, we talked about horizontal gene transfer as messing things up a little bit. So the fact that horizontal gene transfer exists and has existed in essence since the very beginning, then it makes that very base, the root, if you will, of the tree of life, blurry. Okay, so we don't know exactly what that common ancestor was back then, because it's very difficult to trace who came from what and what's going on. The domain Archaea, members of this domain share certain traits with bacteria, but then they share other traits with the eukaryotes. So we've got a problem with this. It's thought that this is our common ancestor and that bacteria became one branch and the other branch ended up diverging into the eukarya and the Archaea domains. In this table, you've got this table in your book, we've got a comparison of the 3 domains of life. So this kind of sums up everything for you guys. It tells about the nuclear envelope, membrane enclosed organelles, the peptidoglycan in the cell wall, membrane lipids, talks about that, RNA polymerase, which is the enzyme that polymerizes RNA, if you will, how protein synthesis is made, whether or not that there's introns, circular chromosome, growth at temperatures that are above 100 degrees Celsius. So characteristics that really define our different domains. Okay, so spend a little bit of time with this. Kind of understand, but don't memorize. Okay, so we talked about nuclear envelope, we talked about organelles, we've talked about the peptidoglycan and the lipids, we talked about circular chromosomes, okay, growth at temperatures above 100, only the members of the domain Archaea fall into this category. The rest of them don't spend time with, okay. So the Archaea, these guys live in very extreme environments. So Archaea is referring to archaic or referring to very old, and it was thought very old bacteria. That's not considered the case anymore because it's pretty widely accepted that this is a separate lineage. So a lot of these Archaea are called extremophiles, meaning that they love living in the extremes. So extreme halophiles love salty environments. Extreme thermophiles really love hot environments. So Archaea tends to live in environments that other organisms can't live in. And they're very well known for that. Methanogens are a type of prokaryotes that are found in the Archaea. These guys live in swamps and marshes and produce methane as a waste product, so very smelly. They're anaerobes, and they can't live in the presence of oxygen. So it's thought that methanogens, as well as other organisms that live in these extreme groups without the presence of oxygen, may give a clue as to how early Earth actually was many, many, many years ago. Okay, so that's it for the Archaea. We're done. So moving on to the domain bacteria. This is most of the prokaryotes. Okay, so very diverse nutritional types in the groups of bacteria. So we've seen that before. Okay, are you guys ready? We're going to go. So again, the domain bacteria is set up in a different way than our kingdom, phylum, so on and so forth. So we're going to look at these groupings that we'll go with the term clade. Okay? So proteobacteria, so we're numbering these. Proteobacteria is the first one. These guys are gram-negative, so we're writing down characteristics of them. The mode of nutrition will be photoautotrophs, chemoautotrophs, and heterotrophs. Metabolic functions, some of these are anaerobic, others are aerobic, so it's kind of dependent. So our proteobacteria is broken up into subgroups, and I do want you to know what these subgroups are. The subgroups are alpha, beta, gamma, delta, and epsilon. Okay, so they're listed here for you. Each of these figures is showing us each of these subgroups, and underneath we've got a genus that is showing us an example. So I want you to know the group, or the major clade, if you will, and I want you to know the subgroups as well as an example. Okay, so example of the alpha proteobacteria is Rhizobium. Nitrosomonas is in beta. Delta is Chondromyces, which is a myxobacterium. Epsilon is Helicobacter pylori, and Helicobacter is the one that makes the ulcers in the digestive system. Diomargarita, part of the gamma, and these can metabolize sulfur. Okay, so some characteristics of each. The alpha proteobacteria, these are most closely related to eukaryotes. Examples are Rhizobium. Another example is Agrobacterium. Both of these help plants. Okay, so, well, in kind of. Agrobacterium can produce tumors in plants, but Rhizobium helps to fix nitrogen and forms a root nodule or a symbiotic relationship with plants. So a legume is a type of plant. Here's our figure. Beta, the example here is a soil bacterium, Nitrosomonas. Gamma, these are the sulfur bacteria such as Chromatium. Pathogens such as Legionella, Salmonella, and Vibrio cholerae. All of these cause disease. That's what a pathogen does. It causes disease. Legionella is Legionnaire's disease. Vibrio cholerae can form cholera, which can be deadly. Escherichia coli falls into this category as well. Delta is the myxobacteria. So myxos referring to mold-like or fungus-like. And epsilon contains the Campylobacter, which can cause blood poisoning, and the Helicobacter, which is the ulcers for the stomach. Okay, here's examples of the other groups. This is all in your book. So chlamydias, these are parasites that live within animal cells. So the genus here is chlamydia, and it's a sexually transmitted disease that can cause blindness. And this is what it looks like. So you're looking here at the arrows. Spirochetes, so this is the next group. These are helical heterotrophs. Several examples here. Treponema, which causes syphilis, and Borrelia, which causes Lyme disease. Treponema and Borrelia. Just know these by genus. Okay, don't worry about the species, just the genus. Okay, so this is Leptospira, so this shows you the spirochete. Cyanobacteria, these guys are photoautotrophs. Here's an example, Oscillatoria. You guys saw this in lab. Gram-positive bacteria, and there's several examples of these. Actinomycetes, which is fungal-like as well. Bacillus anthracis, which causes anthrax. Clostridium botulinum, these you would need to know the genus and the species. Clostridium botulinum causes botulism, which comes from those dented cans. Staphylococcus and streptococcus, which can cause sickness, strep throat. And then mycoplasms are the smallest known cells, and these fall into our gram-positive bacteria. Okay, so when you study these, make sure for each group that you study the characteristics and also the examples. So I would know the examples and be able to possibly do some matching questions for those. Now, our prokaryotes are important. So these play very crucial roles in the biosphere. So we see these organisms everywhere. So if these guys were to disappear, I think life on Earth as we know it would disappear as well. Some of the things that prokaryotes do, and let's get the highlighter going on this one, they can act as decomposers. Okay, so these prokaryotes can help with the cycling of chemicals in the environment itself. So they can act as decomposers to break down dead organisms and also waste products. So when a cow in the pasture relieves themselves, so to speak, it's bacteria that's going to break down that cow pie that's out there. Prokaryotes can help with plant growth. Okay, but prokaryotes can also decrease plant growth, so to speak. So they can decrease the availability of nutrients. So they can actually kind of strip an area of nutrients so that it can be a good thing, it can be a bad thing on both sides. There are several instances of symbiosis that occur with our prokaryotes. So symbiosis is a relationship, so an ecological relationship where two species live in very close contact with each other. You're going to have a very large host, typically, and a smaller symbiont. So those prokaryotes very often form symbiotic relationships with larger organisms. So very common. To talk about the different types of symbiotic relationships, there's mutualism, commensalism, parasitism, and that's it. So mutualism, let's take a look at that. Both the host and the one that's living on the host are going to benefit. So both symbiotic organisms will benefit. So it's a mutual association, so to speak. In commensalism, one organism is going to benefit while the other organism is neither helped nor harmed in any way. And in nature, it's really hard to see examples of commensalism. I can't even think of one to give you, so to speak. Parasitism, so mutualism is very common. Parasitism is very common as well. And this is when you have one organism that is living off of its host. Okay, so parasitism, it's in the parasite's best interest to keep the host alive. If the host dies, then they're going to die too. So a parasite typically harms, but it does not kill its host. Parasites that cause disease are called pathogens. Okay, same for bacteria that cause disease. They're called pathogens. Prokaryotes can have both a beneficial and a harmful impact on humans. Okay, so some are considered to be pathogens, but others have very positive interactions with humans. Our mutualistic bacteria, okay, to look at that particular case, the human intestines have tons of bacteria. So about 500 to 1,000 species of bacteria, multiply that by however many. So that's our local flora that lives within our digestive tract, and it's normal. Okay, so a lot of the food that we can't deal with, that bacteria is going to deal with for us. So it's a good thing. Pathogenic bacteria, on the other hand, is going to cause disease. So one of the examples here is Lyme disease. And Lyme disease is Borrelia, and it's carried by ticks. So it's a very specific bacterium that is carried by a tick. So this is what the tick looks like right there. So these guys are small, little deer ticks. This is what the bacteria looks like, so the little spirochete. And if you've been bitten by the deer tick, in about 70 to 80% of the cases, you get this very characteristic bullseye rash. And that rash is indicative of you having been bitten by a tick that was infected with this particular bacterium, which is very treatable with antibiotics. If you don't treat Lyme disease, then it can work its way into the nervous system. Then you've got a lot of problems. Exotoxins and endotoxins can be released by these pathogenic prokaryotes. Exotoxins are secreted to the outside. Okay, so they're secreted to the outside, and they're going to cause disease even if the prokaryotes that produce them aren't present. Okay, so the bacteria itself no longer has to be present, but the toxin that's been secreted to the outside will do damage to the organism that it's in. Endotoxins are released when the bacteria actually die. So endotoxins are going to stay within the cell. The bacterium has to die in order to release that endotoxin. Okay, so that's pretty cool. We've talked about horizontal gene transfer. You should keep in mind that some pathogenic bacteria are potential weapons for bioterrorism. Anthrax is one of them. Anthrax can be very nasty and kill somebody very quickly. Prokaryotes are very commonly used in research and technology, and in fact, you will use prokaryotes in Bio-6 to do some transformations. We will use E. coli for gene cloning, so we will actually do that in class. Agrobacterium is used to produce transgenic plants. And bacteria now can even be used to make natural plastics. So bacteria can be very beneficial. We know about bioremediation. So that is removing pollutants from the environment, such as oil. I had mentioned before the oil-eating bacteria that can remove the oil from the environment. We can use bacteria to make vitamins, to make antibiotics, and to even make hormones that humans can ingest. Bacteria are being used to produce ethanol from waste. Okay, so that is commonly being used as well. So microbiology deals with the field of our bacteria, and it's a huge field. So it's very interesting, and it might be an area of biology that you want to go into and study a little bit more about. There's so many different fields that you can look at. Food science is one of them. Enology, which is the making of wine. You could work at the CDC and learn how to look at the diseases that these bacteria cause. So many different things that you can do in this particular field. Okay, we are done with this lecture. I hope you've enjoyed it. Pay attention to the characteristics that we've gone over, and keep in mind that for several chapters that we will go through looking at a lot of different characteristics, okay, that place our organisms in our particular categories. So be aware of that.